So throwing a banana peel into a kitchen bin, it, it feels like the most final, uneventful action in the world. You toss it, you close the lid, and as far as you're concerned, the story of that piece of fruit is over.
Right, out of sight, out of mind.
Exactly. But if you were to zoom in, if you could just, like, drop down to a microscopic scale inside that dark, humid plastic container, you would see that the moment the lid closes, an entirely new, incredibly violent, and highly choreographed world wakes up.
Oh, absolutely. It's a complete ecosystem just exploding into action.
Yeah, and welcome to today's deep dive. Our mission for you today is to take you straight into that hidden world beneath our feet and right in our waste bins.
And we are looking at a really wild stack of sources today. They span completely different scientific disciplines.
They really do. I mean, we've got agricultural data on the exact chemical ratios required to make organic matter rot efficiently.
Right, the fundamental biochemistry.
Yeah, and then we've got hardware specs from engineering journals detailing how, like, massive urban compost systems are being wired with the Internet of Things.
Which is wild. And then, as if that wasn't enough, we have papers on the absolute frontier of synthetic biology.
Yes, papers detailing how scientists are preparing to literally genetically rewrite the soil microbiome itself.
It's a fascinating synthesis of disciplines, really, because for most of human history, decay was just, well, something that happened to us.
Right, it was just there.
Exactly. It was a passive background process. But as we comb through these sources, from the fundamental thermodynamics of a compost pile to the deployment of edge computing and methylation-sensitive CRISPR editing, a very clear narrative emerges.
And what is that narrative?
We are taking the oldest biological engine on the planet, the process of rot, and we are actively reverse engineering it so we can control it.
Okay, let's unpack this. Because to understand how we are trying to control this process, we first have to understand the fundamental physics and chemistry of the engine itself, right?
Right, you have to know how the engine runs before you can hack it.
Exactly. And that engine runs on a very specific, unforgiving type of fuel mixture. In the composting world and in the sources we are looking at from Cornell University and various industrial guides, everything revolves around one metric.
The carbon to nitrogen ratio.
Right. The CN ratio.
And that ratio is the absolute bedrock of decomposition. The consensus across all the agricultural and waste management data is incredibly strict on this.
Yeah, it's not just a suggestion.
No, not at all. The ideal sweet spot for rapid hot composting, the kind that breaks down organic matter efficiently, is a ratio between 25:1 to 30:1.
Okay, so that is 25 to 30 parts of carbon for every single part of nitrogen.
Exactly.
And when you look at the raw materials we actually throw away, the numbers fluctuate wildly. Composters usually categorize materials into browns, which are your carbon-heavy materials, and greens, which are your nitrogen-heavy ones.
Right, and it's helpful to look at the extremes here to understand the scale.
Totally. Cardboard, for example, is basically just pure structural fiber. It has a massive CN ratio. It sits anywhere from 400:1 all the way up to 560:1.
Massive amounts of carbon.
Yeah. And then straw, which farmers use constantly, is about 75:1 up to 85:1. Those are your quintessential browns.
And then you have the greens.
Right. Fresh grass clippings from a lawnmower, those sit right around 15:1, maybe 25:1.
Which is getting closer to that ideal sweet spot.
Yeah, but then you get to the heavy hitters, like blood meal, which is an animal byproduct. That stuff is incredibly nitrogen dense at roughly 3:1.
Wow. And to understand why that 25:1 ratio is the universal target, you have to look at the literal biological mechanisms of the microbes doing the work. You can't just, throw everything in a pile and hope for the best.
Right, because they're living organisms.
Exactly. The microbes have specific metabolic demands. They use carbon primarily as an energy source. It is the fuel that powers their respiration.
So they eat the carbon to get energy to move and live.
Yes. When they consume carbon-rich materials, they oxidize that carbon, and a significant portion of it is actually lost to the atmosphere as carbon dioxide.
Okay. And the nitrogen?
Nitrogen, on the other hand, is not burned for fuel. It is the fundamental building block they use to construct amino acids, proteins, and genetic materials so they can actually reproduce.
Now wait, let me push back on this, because the logic feels a little counterintuitive to me.
Okay, how so?
Well, if nitrogen is the building block, if it is the raw material required for the microbes to build cell walls and replicate, wouldn't a pile of pure grass clippings
Mhm.
or pure nitrogen-dense material just cause the microbial population to explode incredibly fast?
I see what you're saying.
Like, why are we adding the cardboard at all? If
Yeah.
I want a fire to burn hotter and faster, I don't throw slow-burning, dense logs on top of pure rocket fuel. It feels like adding carbon is just watering down the system.
What's fascinating here is that microbial metabolism has very strict structural limits. And your fire analogy actually perfectly illustrates the problem, just not in the way you might think.
Really? Tell me how.
Well, a fire needs oxygen to burn fuel, right? If you flood a fire with fuel but cut off the oxygen, the fire chokes. Microbes operate under similar constraints.
Okay, I'm following.
If the CN ratio drops below 20:1, meaning there is an abundance of nitrogen but a severe lack of carbon fuel, the microbes literally do not have the metabolic energy required to process and assimilate all that nitrogen.
Oh, wow. So they can't just absorb it passively.
No, building proteins takes energy. Without enough carbon to burn for that energy, the assembly line just breaks down.
So they're essentially sitting in a warehouse full of construction materials, but the power to the factory has been cut off.
That's a great way to put it.
They have the bricks, but no electricity to run the cranes.
Exactly, and the consequences of that power outage are severe. We can look at a foundational study by Tikia and Tam from the year 2000, which tracked exactly what happens to that excess unprocessed nitrogen.
Because it doesn't just sit there, right?
Right, it doesn't just sit there benignly in the pile. Because the microbes can't assimilate it, it undergoes a chemical conversion and escapes the system as ammonia gas, NH3.
Oh, that sounds bad.
It is. The data in that study showed that when the starting ratio is too low, a staggering 20% to 35% of the total initial nitrogen in the pile can be lost to the atmosphere through volatilization.
Which means it literally evaporates away into the air.
Exactly, it's just gone.
And anyone who has ever smelled a poorly managed compost pile knows exactly when this is happening, because ammonia gas has that incredibly sharp, choking, terrible odor. It smells like a dirty litter box.
It does. It's unmistakable. And beyond the smell, think about the sheer inefficiency of that. Nitrogen is the most valuable nutrient in the entire compost mixture.
Right, it's what plants need to grow.
Yes, it is the gold that farmers are trying to harvest. By leaving out the cardboard, by failing to provide the carbon energy, you aren't speeding up the creation of fertilizer, you are actively destroying it.
Wow.
You're wasting massive amounts of nutrients and creating a local air quality crisis. The carbon is the required energy tax the microbes charge to secure the nitrogen into a stable form.
That shifts the whole perspective. The cardboard isn't filler, it's the battery power.
Precisely.
So let's say you get the ratio perfectly dialed in, you hit that 25-to-1 mark. The microbes have the perfect balance of fuel to burn and materials to build with. The population explodes.
Right. Yeah.
As they consume the carbon, as you mentioned, they are oxidizing it. And the byproduct of billions of microscopic organisms constantly oxidizing carbon is heat.
A tremendous amount of heat. When a compost pile is properly structured, the sheer density of microbial respiration pushes the internal environment into what is called the thermophilic stage.
The thermophilic stage.
Yeah, this is a critical biological phase where the internal temperatures rise to between 40 and 60 degrees Celsius. That's roughly 104 to 140 degrees Fahrenheit.
Wait, really? That is hot enough to cause a severe burn if you were to reach your bare hand into the center of the pile.
It absolutely is. And that intense heat serves a vital evolutionary and agricultural function. Sustaining those thermophilic temperatures for several consecutive days is the primary mechanism for sanitation.
Sanitation, like cleaning the dirt.
Essentially, yes. The heat physically denatures the proteins inside thermosensitive pathogens like Salmonella and E. coli. It destroys the cell structures of fly larvae, and it effectively cooks the embryos inside weed seeds, rendering them sterile.
Oh, wow. So the heat purifies the waste.
Yes, but it is an incredibly delicate thermodynamic balancing act.
Because if it gets too hot, the system crashes.
Exactly.
The sources indicate that if the internal temperature exceeds 65 degrees Celsius, the beneficial thermophilic microbes themselves begin to die off.
Right. Their own enzymes denature at that point.
And the decomposition process just stalls out completely, and the pile goes inert until it cools down enough for the population to slowly recover.
Which introduces the massive challenge of thermal management. The heat generated by the microbes is constantly moving and escaping. It's basic thermodynamics operating in a pile of rotting vegetables.
Yeah, the literature breaks down 3 primary avenues of heat loss. First, there is conduction, Right.
Yes. Conduction happens because the outer edges of the compost pile are in direct physical contact with the cooler ambient air, and heat transfers directly into that cooler mass.
Right, which is why the outer shell of a compost pile is always cooler and less decomposed than the core.
Exactly. Then you have convection. As the air inside the center of the pile heats up, it expands and rises, escaping out the top and drawing cooler, oxygen-rich air in from the bottom.
Like a chimney effect.
Yes, exactly like a chimney. But crucially, that rising warm air carries a massive amount of water vapor with it. This is called latent heat loss.
Latent heat loss.
Right. The energy required to convert liquid water in the compost into vapor is enormous, and as that vapor escapes, it strips thermal energy out of the system.
Okay, and the third one.
Finally, there's simple radiation, the pile emitting infrared electromagnetic warmth out into the environment.
And this is where the physical structure of the pile becomes so important, specifically moisture content. The guides state that you want moisture levels hovering right between 50% and 60%.
Right.
If you squeeze a handful of compost, it should feel like a damp sponge. Not dripping, but not dry either. And the reason for that specific moisture level is that water acts as a thermal flywheel.
Yes, water has an exceptionally high specific heat capacity. It requires a lot of thermal energy to raise the temperature of water by even a single degree.
So it absorbs the shock.
Exactly. In a compost pile, that moisture acts as a buffer. When microbial activity spikes and generates a massive surge of heat, the water absorbs it, preventing the temperature from immediately skyrocketing past that lethal 65-degree threshold.
And when it cools down.
When the microbial activity slows down, the water slowly releases that stored heat back into the pile, preventing the temperature from plummeting. It stabilizes the microscopic environment.
Okay, so let's zoom out for a second. We established that a healthy compost pile is essentially a slow-burning microscopic fire.
Yes.
It's strictly governed by a 25-to-1 chemical ratio, buffered by specific water weights, and heavily dependent on airflow to manage convective heat loss.
Correct.
Now, for a backyard gardener, managing this is just a weekend hobby. You turn the pile with a pitchfork, you feel the heat radiating off it, and if it smells bad, you throw some dry leaves on it.
Very hands-on.
Yeah. But what happens when you scale this up? Because the sources we are looking at aren't focused on backyard bins. They are looking at massive, high-density urban environments.
Whole different ballgame.
How do you monitor this invisible biological fire when you have thousands of pounds of waste sitting in the courtyard of a luxury apartment complex? You can't exactly have a superintendent out there sniffing the garbage every 6 hours.
No, and you can't rely on manual temperature probing either because it is incredibly labor-intensive and prone to human error. This is where we see the ancient biological process of composting colliding directly with modern computer science.
It's such a wild combination.
It really is.
Wow.
Historically, humanity has always understood the immense value of composting. One of the MDPI papers provides great context here, noting that the Aztecs engineered highly complex, large-scale composting systems.
Really, the Aztecs.
Yeah. They used human latrines and bat guano to maintain the fertility of their chinampas, the artificial islands they used for agriculture. They scaled the process through sheer manual labor.
Wow.
But today, in a modern urban matrix, we require continuous precision without the labor overhead. We are forced to bring the compost heap into the digital age.
Which brings us to this fascinating study from the MDPI Internet of Things journal. The researchers designed and deployed a network of compost bins in residential apartment complexes.
Yeah.
And these are massive units.
How massive are we talking?
We are talking about bins with up to an 800-kilogram capacity. Over 1,700 pounds of rotting organic matter sitting in close proximity to where people live and sleep.
Oh, so the margin for error is absolutely zero.
Exactly. You cannot have an accidental ammonia leak or a sudden spike in noxious gases. To prevent that, they equip these massive bins with what they call sensor hubs.
The architecture of these sensor hubs is a masterclass in edge computing and low-power IoT deployment. Rather than just sticking a digital thermometer in the dirt, they built a highly localized data processing center right on the bin.
Oh, so it's doing the thinking right there.
Yes. The primary data collection is handled by an Arduino Uno microcontroller. The Arduino interfaces directly with the physical sensors, temperature probes, gas sensors, moisture meters, and collects the raw analog data. It then transmits that data via a UART port to a Raspberry Pi.
Let's define what we mean by edge computing here, because it's a crucial concept for why this network actually functions in the real world.
Good idea.
In a traditional cloud computing model, a sensor takes a reading, and immediately sends that raw data over the internet to a centralized server, the cloud, which is all the math and analysis.
Right, but sending constant wireless transmissions requires a lot of electricity.
Exactly. If you have a battery-powered sensor constantly transmitting raw data to a cloud server 50 miles away, the battery will drain in a matter of days.
And you don't want to be changing batteries in compost bins every week.
No, you don't.
Yeah.
So in an edge computing model, the computation happens at the edge of the network, right where the data is collected. The Raspberry Pi on the compost bin acts as a local manager.
It filters everything.
Right. It takes the raw data from the Arduino, processes it, analyzes it for anomalies, and only transmits a signal when it is strictly necessary or at predetermined, highly spaced out intervals.
And when it does transmit, it uses a specific type of network. I noticed in the hardware breakdown they utilized a LoRaWAN probe, specifically the KO10 model.
Yes, LoRaWAN. That stands for Long Range Wide Area Network.
Which is such a cool technology.
It is. It uses sub-gigahertz radio frequency bands. It cannot send a lot of data like you couldn't use to stream video, but it can send tiny packets of text data over very long distances.
And it penetrates well.
Right, exactly. It can penetrate right through dense materials like wet compost and concrete walls. And because the data packets are so small and the frequency is so low, the power draw is microscopic. The battery in that KOU10 probe can last up to 7 years.
That 7-year battery life completely rewrites the economics of urban waste monitoring. It means a city municipality can deploy hundreds of these sensors across a district and basically forget about hardware maintenance for the better part of a decade.
Which is huge for city budgets.
Massive. And these hubs are tracking an incredibly comprehensive array of vital signs. They monitor the ambient air temperature, the internal compost temperature at various depths, the exact moisture percentage, the pH levels, and crucially, they're sniffing for very specific volatile gases.
Right, the gas sensors are the early warning system. They monitor for ammonia, NH3, which, as we established earlier, immediately tells the system that the CN ratio has dipped too low and nitrogen is volatilizing.
And what else are they looking for?
They also monitor for methane, CH4, and hydrogen sulfide, H2S.
Hydrogen sulfide. That is the chemical compound responsible for the classic rotten egg smell.
Yeah, it's terrible.
When methane and hydrogen sulfide spike in the data, it tells the researcher something very specific about the physical architecture of the pile. It means the pile has gone anaerobic.
Because the microbes have run out of oxygen.
Exactly. Aerobic respiration, which produces the heat and the carbon dioxide we want, requires oxygen. As the microbes multiply and consume the waste, they rapidly deplete the oxygen trapped in the pore spaces of the compost.
So they basically suffocate themselves if we don't intervene.
Precisely. If the pile isn't turned, or if the moisture level gets too high and water fills those pore spaces, oxygen can no longer penetrate. The aerobic microbes die or go dormant, and anaerobic bacteria take over.
And they are the ones making the bad smells.
Yes. These anaerobic bacteria ferment the organic matter without oxygen, and their metabolic byproducts are not heat and CO2, but methane, hydrogen sulfide, and organic acids that drop the pH.
So instead of a maintenance worker having to walk out to the bin, stick a massive thermometer deep into the pile, and take a deep breath to see if it smells like rotten eggs, the Raspberry Pi is continuously monitoring the invisible gas emissions.
It's a fully automated watchdog.
Yeah. If the methane concentration hits 2% of the baseline value, the edge device immediately recognizes the anaerobic shift. It sends a tiny data packet over the LoRaWAN network to a supervisor's phone with an alert saying, bin number 4 at the West Complex has gone anaerobic. It needs to be physically turned to reintroduce oxygen into the system.
It is a phenomenal leap in logistical efficiency. However, the researchers in the MDPI study recognized a fundamental limitation in using physical probe sensors, and their solution to this limitation is one of the most intriguing parts of the paper.
Okay, yeah.
They introduced thermal imaging into the monitoring matrix.
I really want to dig into this because when I was reading the methodology, this detail completely stopped me in my tracks.
Oh, I bet.
I looked up the equipment list. They deployed a FLIR E8 Pro thermal camera to monitor the bins. Now, I checked the current market price for that specific piece of hardware. That camera cost roughly 323,000 Indian rupees.
Wow.
That is almost 4,000 US dollars. Contrast that with a standard thermistor probe, which costs maybe $3 or $4. My immediate reaction was, why on earth would anyone point a $4,000 military-grade camera at a pile of garbage? Isn't a cheap digital thermometer pushed into the dirt enough to tell you if it's hot?
It comes down to the problem of spatial variance.
Visual variance.
Right. A physical probe, no matter how accurate, only provides a single, highly localized data point. When you push a thermometer into an 800-kilogram pile of compost, it is only measuring the temperature of the exact cubic inch of material that the metal tip is physically touching.
Right. It is completely blind to everything happening 6 inches to the left.
Exactly. And a compost pile is an incredibly heterogeneous environment. The structural density varies. The moisture pools unevenly. The airflow creates channels.
And messy.
Very messy. You could have a localized pocket of perfectly aerated, highly active material burning away at a healthy 60 degrees Celsius right next to a dense, compressed pocket of cold, wet, anaerobic sludge.
And if your probe happens to be in the hot spot.
Exactly. If your physical probe happens to be pushed into the hot zone, the data tells you the pile is perfectly healthy, completely missing the anaerobic failure happening right next to it. A thermal camera solves this by measuring the infrared radiation emitted across the entire exposed surface area of the pile simultaneously.
But wait, the camera can't see into the pile, right? Infrared doesn't penetrate soil. It's only looking at the surface temperature, and the surface is constantly being cooled by the ambient air. How does surface temperature tell you what's happening deep inside the core?
Well, it's not the absolute temperature that they are looking at. It is the variance.
Okay, explain that.
The software takes the thermal image and converts the infrared data into a grayscale histogram, which is basically a mathematical distribution of the temperatures across the entire surface area. By analyzing that histogram, the algorithm calculates the thermal variance.
I see.
If the variance is low, meaning the surface temperature is relatively uniform across the entire bin, it mathematically indicates that the internal biological activity is also uniform. The heat is rising evenly.
That makes total sense. And if the variance is high?
If the histogram shows massive spikes and valleys, hotspots, right next to cold spots on the surface, it definitively proves that the internal structure has compromised.
Wow.
The high variance on the histogram reveals that there are uneven pockets of decomposition, blocked airflow, and potential anaerobic zones underneath. The algorithm can assess the structural health of the entire 800-kilogram mass in a fraction of a second without ever disturbing the material.
So you are paying for a macro-level structural health check that a single-point probe simply cannot provide.
Exactly. It's worth every penny when you're managing that much waste.
That is brilliant. It's using surface-level thermodynamics to infer subterranean biological geometry. But what's great about this study is that they aren't just adding expensive hardware. They are also using the massive data sets they're generating to find ways to actively cut costs.
Right, through the gas correlations.
Yes, they ran a correlation analysis on the gas emission data, right?
Yes, they did. Over months of continuous monitoring, capturing thousands of data points a day, the researchers mapped out the behavioral relationship between the different gases. What they found was a very high, statistically significant correlation between hydrogen sulfide levels and methane levels.
Meaning they behave identically under the same conditions. When the oxygen runs out and the pile goes anaerobic, the methane spikes and the hydrogen sulfide spikes in tandem, they track together.
Precisely. Which means from a hardware engineering perspective, capturing both data points is totally redundant. You do not need both sensors to trigger the anaerobic alert.
Oh, so you can just drop one.
Exactly. In future iterations of these commercial bins, manufacturers can confidently eliminate either the methane or the hydrogen sulfide sensor entirely. This drops the manufacturing cost, reduces the required processing power of the edge device, and extends the battery life even further, all without losing any operational fidelity.
That is the true power of applying big data analytics to biological systems. You map the redundancies and you engineer them out.
It's elegant.
It really is. Okay, so the IoT infrastructure is in place. The sensors are pinging us with incredibly precise data. We know exactly when the ammonia levels are creeping up. We know exactly when the methane is spiking. And we know the thermal variance of the pile.
We have eyes on the fire.
Yeah. But to understand why those specific gases dictate the entire success or failure of agriculture, we have to follow those nutrients out of the compost bin and into the earth. We have to look at the underlying economy of the soil itself. We need to talk about the nitrogen cycle.
This is a vital transition. We are moving from the physics of waste management into the fundamental biochemistry of global agriculture.
Right.
Our sources from the California Department of Food and Agriculture and the deep dive articles from the Progressive Crop Consultant detail this dynamic beautifully. They describe soil not as dirt, but as a highly volatile economic market where nitrogen is the primary currency.
It's all about the nitrogen.
It is a delicate, continuous dance of nitrogen moving in and out of what scientists call soil organic matter, or SOM.
The texts describe two massive opposing forces constantly warring within the soil: mineralization and immobilization. Let's break these down because everything that grows on Earth depends on the balance between these two forces.
Well, let's start with mineralization. This is the process every farmer, gardener, and ecologist wants. When you spread compost on a field, that compost is full of organic nitrogen.
Organic nitrogen, right?
It is locked up inside complex proteins, amino acids, and the DNA of dead microbial cells. Plants, generally speaking, cannot eat organic matter. A corn plant cannot absorb a chunk of protein through its roots.
It's too complex.
Yes. It requires nitrogen in a simple inorganic form, specifically ammonium NH4 plus N5, or nitrate NO3, which dissolves cleanly into the soil water.
So the compost is useless to the plant until it is processed.
Completely useless. The microbes in the soil have to act as the external digestive system for the plants.
That's a great image.
During mineralization, the microbial workforce consumes that complex organic matter, breaks the chemical bonds, utilizes the carbon for energy, and excretes the excess nitrogen out into the soil as inorganic ammonium. They are literally mineralizing the organic material, turning it into liquid fertilizer that the plants can immediately absorb.
But then you have the opposing force, immobilization, which is the exact reverse of that process.
Sadly, yes.
Instead of releasing inorganic nitrogen into the soil microbes actively pull that liquid mineral nitrogen out of the soil solution, absorbing it into their own microscopic bodies to build their own cells. They lock it away, essentially hoarding the fertilizer and starving the plants.
Correct. And the trigger that dictates whether a field of soil undergoes mineralization or immobilization is the exact same metric we discussed with the compost pile. The CN ratio.
The magic dividing line.
Yes. In the soil, a CN ratio of 20:1 is the critical threshold. If the organic matter in the soil has a ratio below 20:1, it contains more nitrogen than the microbes need for their own cellular reproduction.
So there's a surplus.
Right. They consume it, take what they need, and excrete the excess as plant-available ammonium. Mineralization occurs. But if the ratio spikes above 20:1, the dynamic violently reverses.
Okay, let me try an analogy here to see if I'm really grasping the mechanics of this, because this seems like a terrifying scenario for a farmer.
Let's hear it.
Let's say a farmer plows a massive amount of high-carbon crop residue into their field, say thousands of pounds of dry wheat straw, which we know has a ratio of 80:1. This massive influx of carbon drops into the soil, immobilization kicks in. Is it essentially a microscopic bank run? The microbes in the soil see this massive deposit of carbon energy. They wake up, they start burning the carbon, and they desperately want to multiply. But to build new cells, they need nitrogen. The straw doesn't have any.
Right, it's just carbon.
So the entire microbial population rushes the bank. They aggressively absorb all the free-floating liquid cash, the available inorganic nitrogen that was already in the soil, locking it up in their own bodies and leaving the plants completely broke.
If we connect this to the bigger picture of agricultural economics, your bank run analogy is perfect.
Wow, that's wild.
The microbes have a competitive advantage over the plant roots. They are smaller, more numerous, and faster. When that massive influx of carbon hits the system, the microbes will rapidly drain the soil of every drop of available inorganic nitrogen. And this exact phenomenon highlights why organic farming is such a uniquely massive and complex challenge compared to conventional farming.
Walk me through that difference. Why is it harder for an organic farmer?
In conventional agriculture, a farmer bypasses this entire biological economy. They apply synthetic nitrogen fertilizer directly to the field.
Right, skipping the microbes entirely.
Yes, that synthetic fertilizer is manufactured in a chemical plant, usually via the Haber-Bosch process, and it is already in an inorganic plant-available form. It's like a helicopter dropping pallets of cash directly into the plant's bank account. It doesn't need to be digested.
But an organic farmer operates under a completely different set of rules. They are not allowed to use synthetic fertilizers. They rely entirely on organic amendments, compost, animal manure, cover crops, and blood meal.
Which means every single ounce of nitrogen they apply must be mineralized by the microbes before the crops can use it.
Exactly. The organic farmer is completely 100% at the mercy of the microbial bank tellers. If an organic farmer miscalculates the CN ratio of their compost and accidentally spreads material that is 35:1 instead of 15:1, They will trigger a massive immobilization event.
And that's not their only problem.
Right. Furthermore, they are at the mercy of the environment. Microbes are temperature dependent. In the early spring, when the soil is still cold, microbial metabolism is incredibly slow. The bank tellers are basically asleep.
Yep.
An organic farmer can have fields loaded with organic compost, but because the soil is cold, mineralization crawls to a halt. Their crops will literally stunt and turn yellow from nitrogen starvation while being completely surrounded by locked-up nutrients. The fertilizer is there, but the vault door is closed.
And the soil structure itself changes the thickness of that vault door. The Progressive Crop Consultant article got incredibly detailed about how the physical architecture of the dirt dictates the speed of this economy.
Yeah, that part was fascinating.
They distinguished between two types of organic matter in the soil: particulate organic matter, or POM, and mineral-associated organic matter, or MAOM.
This is a crucial physical distinction. Soil is not just a homogeneous chemical soup. It has physical architecture, sand, silt, and clay. Particulate organic matter, the POM, is exactly what it sounds like. It's tiny bits of decomposing leaves, root hairs, and plant stems suspended in the soil. It is physically accessible. A microbe can easily swim up to a piece of POM, attach itself, release enzymes, and begin mineralizing it. It is highly labile, meaning it breaks down quickly.
So POM is the loose cash sitting on the teller's counter. Easy to grab.
Precisely. But mineral-associated organic matter, MAOM, is fundamentally different. This is organic matter that has been broken down to a microscopic level and has actually formed chemical bonds with the microscopic mineral particles in the soil, particularly clay.
Ah, clay.
Yeah, clay particles have an incredibly high surface area and carry a negative electromagnetic charge. They attract and chemically bind to organic molecules, forming a microscopic physical barrier around them.
So the clay literally acts as a physical vault, locking the organic matter away from the microbial enzymes.
Yes. Soils with a high clay content have significantly more MAOM than sandy soils. Now, from an environmental perspective, this is fantastic because MAOM is how soil sequesters carbon long-term, keeping it out of the atmosphere.
Right, it's great for the climate.
But from an agricultural perspective, it makes farming incredibly difficult. The mineralization rate, the speed at which the microbes can pry that nitrogen out of the clay vault and hand it to the plants, is incredibly slow and notoriously hard to predict.
It is mind-boggling when you lay it all out. To successfully grow a field of organic crops, a farmer has to calculate the exact chemical CN ratio of their compost, cross-reference it with the ambient soil temperature, factor in the moisture levels, and adjust the entire equation based on whether their soil is sandy and full of accessible POM or clay-heavy and locked up in MAOM.
It's crazy.
It is multivariable calculus played out in real time with billions of invisible variables just to grow a head of cabbage.
Which brings us to the ultimate realization driving the next generation of biological science. For millennia, humanity has been forced to play this incredibly complex game of multivariable calculus, passively hoping the microbes behave the way we want them to.
Hoping being the key word.
But the cutting edge of science is no longer satisfied with just monitoring the game. They're preparing to rewrite the rules entirely.
Oh, yes.
Here's where it gets really interesting. This is where we leave the macro world of the compost bin. We bypass the chemical economics of the soil, and we drill straight down into the very DNA of the microbes themselves. We're talking about synthetic biology.
The sources curated from the Scoop.it synthetic biology database show us that we are standing on the precipice of a total paradigm shift. To fully grasp what scientists are attempting to engineer, we first have to understand the baseline reality of how plants and microbes interact.
Okay, set the stage for us.
For the last 450 million years, plants and soil microbes have been locked in a relentless evolutionary dance. They have co-evolved along what the scientific literature terms a friend-foe continuum.
The friend-foe continuum. I love this concept. It means that a microbe isn't just inherently good or inherently bad in a vacuum. It is constantly negotiating with the plant's sophisticated immune system.
Exactly. The molecular mechanisms of infection and symbiosis are shockingly similar. A plant pathogen is just a microbe that has evolved to breach the plant's defenses, extract nutrients, and offer absolutely nothing in return.
A pure parasite.
Right. And that triggers a massive immune response from the plant to kill the invader. But a mutualist, a beneficial nitrogen-fixing bacteria, has evolved incredibly sophisticated strategies to actively evade or artificially suppress that exact same immune system.
It's like hacking the firewall.
Yes. They secrete specific signaling molecules that essentially tell the plant, don't shoot, I bring gifts. They negotiate a symbiotic treaty, trading the nitrogen they fix for the carbon sugars the plant produces through photosynthesis.
And when you look at the lengths these organisms go to in order to maintain that treaty under extreme environmental stress, it feels like science fiction. The Scoop Out or 8 papers highlighted a study on Clostridium bacteria living in the soil of flooded rice paddies, and the mechanics of this blew me away.
That study is incredible.
So a flooded rice paddy is anoxic. The water cuts off the oxygen supply to the soil, but the rice plant needs to survive, so its roots actually leak tiny amounts of oxygen into the surrounding mud.
Just enough to stay alive.
Right, and that leaked oxygen reacts with the iron in the soil, causing it to oxidize and form these microscopic rigid iron plaques coating the surface of the rice roots.
It creates a localized, rust-coated microenvironment around the root. And what the Clostridium bacteria have evolved to do with those iron plaques is an absolute marvel of biochemical engineering.
Okay, let me see if I have this right. The bacteria swim up to the root, attach themselves to the iron plaque, and essentially use it as a microscopic electrical outlet. They plug into the iron so they can recharge their internal batteries and pump massive amounts of nitrogen back into the root.
That is a highly evocative visual analogy, but if we break down the actual biochemistry, it is stunningly accurate. In the scientific terminology of the paper, the bacteria use the oxidized iron plaques as terminal electron acceptors.
Okay, we need to translate that. What is a terminal electron acceptor, and why does a bacteria need one to survive?
Every living cell, from a bacteria to a human, generates energy by moving electrons around. When a bacteria ferments organic matter in an environment without oxygen, it creates a massive internal buildup of electrons. You can think of it as internal redox pressure.
Like pressure in a pipe.
Yes, the bacteria has all these excess electrons flying around, and it needs to dump them somewhere to keep its metabolic engine turning. If it has nowhere to dump them, the engine stalls.
Ah, I see.
In a normal, low-energy environment, it is forced to dump those electrons internally into a very low-yield, inefficient metabolic pathway called solventogenesis. It barely generates enough energy to stay alive, let alone fix nitrogen, which is an incredibly energy-intensive process.
But then it finds the iron plaque on the rice root.
Yes, the iron in the plaque is oxidized, meaning it is hungry for electrons. When the Clostridium bacteria physically attaches to the plaque, it suddenly has an external grounding wire. It can dump all of that built-up internal electron pressure directly into the iron.
It discharges the battery.
Exactly. And by relieving that internal electron pressure, the bacteria can immediately shut down the inefficient solventogenesis pathway and redirect all of its internal carbon and electron flow into a high-yield, hyper-efficient pathway called acidogenesis.
And what does that do?
This metabolic shift dramatically increases the generation of ATP adenosine triphosphate, which is the universal energy currency of the cell. Suddenly, the bacteria is flush with ATP. And because nitrogen fixation requires massive amounts of ATP, this sudden energy wealth allows the Clostridium to supercharge its nitrogen production, flooding the rice plant with the exact nutrients it needs to survive the flood.
That is profound. The plant inadvertently builds the electrical infrastructure the bacteria plugs in, radically alters its entire internal metabolic pathway, generates a surge of ATP, and pays the plant rent in the form of pure nitrogen. Is a perfect closed-loop survival mechanism.
And that is just one example of how these ecosystems adapt under stress. The literature highlighted another study that shifts the focus from plant microbe interactions to microbe interactions.
Oh, this one was amazing too.
They looked at an environment suffering from severe saline alkali stress. This is soil that is incredibly salty and highly alkaline conditions that destroy the cellular machinery of most organisms. In this environment, they studied a specific nitrogen-fixing bacteria called Mesorhizobium, which lives inside the protective root nodules of leguminous plants.
But even inside the protective nodule, the salt stress is too much, right? The Mesorhizobium begins to break down.
Yes, the osmotic stress causes massive cellular damage. Specifically, the researchers found that the bacteria's branched-chain amino acid synthesis pathway and its TCA cycle, which is the core engine for generating energy, become critically compromised.
So it's failing at a fundamental level.
The Mesorhizobium literally loses the ability to manufacture vital metabolites, specifically isopropylmalate and succinic acid. Without those chemicals, its energy production stops. It begins to starve to death inside the root nodule.
But the Mesorhizobium isn't the only bacteria living in that nodule. There are other species, non-rhizobial endophytes, that share that space. And the researchers focused on one specific neighbor, a bacteria called Bacillus siamensis.
Right.
Now, my initial read on this was to call the bacillus a microscopic paramedic, because when the Mesorhizobium starts to die, the bacillus kicks into gear, synthesizes the exact missing chemicals, the isopropylmalate and succinic acid, and pumps them over to the dying bacteria to keep it alive.
It looks like a rescue mission.
Yeah, but calling it a paramedic implies altruism, and evolution isn't altruistic. Why does the bacillus spend its own precious limited energy to save a different species of bacteria in a starvation environment?
That is a phenomenal question, and it cuts to the core of evolutionary game theory. It is not altruism. It is a desperate act of mutual survival. The bacillus cannot fix nitrogen. It relies entirely on the plant host for its nutrients. But the plant host relies entirely on the Mesorhizobium to fix the nitrogen required to keep the root nodule alive. If the Mesorhizobium dies, the nitrogen supply stops, the plant tissue necrosis, the root nodule collapses, and the bacillus loses its home and dies along with it.
It's a house of cards. If one pillar falls, the whole ecosystem collapses.
Exactly. So the bacillus has evolved an emergency response protocol. Under severe salt stress, it upregulates its own internal production of those specific metabolites, generating a surplus, and deliberately crossfeeds them to the Mesorhizobium,
It acts as an external life support system.
keeping the nitrogen fixer alive so the entire microecosystem doesn't collapse. It is a masterclass in ecosystem resilience.
And understanding that deep molecular resilience is the key to everything that comes next.
Hmm.
Because we are no longer satisfied with just watching the bacillus synthesize succinic acid under stress. We are entering an era where we intend to program it to do so on our command.
That's the leap.
The synthetic biology tools highlighted in these sources are terrifyingly precise. We have to talk about Thermocast9.
This raises an important question about how we edit life. Thermocast9 represents a massive leap forward from the early days of CRISPR genome editing. Standard CRISPR-Cas9 allows scientists to act like molecular scissors, cutting a specific sequence of DNA to turn a gene off or insert a new one. But Thermocast9 is significantly more advanced. It is a methylation-sensitive base editor.
Okay, we need to dive into epigenetic methylation here. What does it mean for the tool to be methylation sensitive?
Epigenetics is the system cells use to control which genes are active without actually altering the underlying sequence of the A, C, T, and G base pairs in the DNA. One of the primary epigenetic mechanisms is DNA methylation, where the cell attaches a tiny chemical tag a methyl group to a specific piece of DNA.
Like a Post-it note on the blueprint.
That's a great way to think about it. That tag essentially acts as a volume knob. It can muffle a gene, turning its expression down, or silence it completely. It's how an organism adapts to its environment dynamically. Thermocast9 is engineered to read those methyl tags. It doesn't just look for a sequence of code. It looks for the chemical context of that code.
Yeah.
It can target and edit genes specifically based on whether they are methylated or unmethylated.
Meaning we can target the dynamic, environmentally responsive switches within the microbes. We aren't just altering their static blueprint. We are hacking their real-time operating system.
Yes.
But a massive challenge in synthetic biology has always been delivery. You can edit a microbe perfectly in a sterile lab, but how do you deliver those genetic upgrades or deliver specific engineered enzymes to billions of wild bacteria in a muddy, chaotic field?
Right, it's a huge logistical hurdle.
The literature points to a biological delivery mechanism called extracellular vesicles, or EVs.
For a long time, microbiologists observed these tiny membrane-bound bubbles, these extracellular vesicles, budding off from bacteria, and assumed they were essentially microscopic trash bags, just a way for the cell to jettison waste.
Just floating garbage.
Exactly. But recent multi-omics studies, which are studies that simultaneously sequence the DNA, RNA, proteins, and metabolites of an entire environment, have completely written our understanding of UVs. In aquatic environments and in the liquid films within soil, EVs act as a massive, invisible, high-speed logistics network.
They aren't trash bags, they're Amazon delivery trucks.
Yeah.
What exactly are they shipping across the microbiome?
They are shipping highly functional biological cargo. The multiomics data proves that these vesicles are packed with amino acids, complex disaccharides, carbohydrate-active enzymes, and critical signaling molecules. The researchers observed a phenomenon where certain strains of bacteria that were amino acid oxytrophic, meaning they possess a genetic mutation that stripped them of the ability to synthesize their own essential amino acids, were able to survive perfectly well in the wild.
How? If they can't make the amino acids, they should die.
They survive by constantly scavenging extracellular vesicles secreted by other healthy bacteria. The EVs act as an externalized shared reservoir of functional traits. If a bacteria is lacking a specific enzyme, it doesn't need to evolve it. It can just absorb a passing vesicle that contains it.
And the ultimate vector for sharing traits isn't just vesicles, it's viruses. The papers detail the role of phages, which are viruses that specifically infect bacteria.
Oh, phages are incredible.
When I think of a virus, I think of a biological assassin. It injects its DNA, hijacks the cell, replicates until the cell explodes, and moves on. But the sources suggest phages are actually the primary engine for evolutionary upgrades through horizontal gene transfer.
Phages are incredibly prolific, but they do not always kill their host immediately. Often, a phage will inject its genetic material into a bacteria, and that viral DNA will silently integrate itself into the host's genome, lying dormant. This is the lysogenic cycle.
So it's just hiding.
Right. Eventually, an environmental trigger will cause the viral DNA to activate. It forces the host to build hundreds of new phages, and then it bursts the cell. But biology is messy. When those new phages are being assembled, they frequently make mistakes. They accidentally package chunks of the host bacteria's DNA inside the viral shell alongside their own.
So when that newly assembled phage floats away and infects the next bacteria?
It injects that stolen bacterial DNA into the new host. This is horizontal gene transfer. A phage can accidentally carry a gene for antibiotic resistance, or a gene for extreme heat tolerance, or a gene that vastly improves nitrogen fixation, and forcefully inject it into a completely different species of bacteria on the other side of the field.
It's like a network upgrade.
Exactly. Phages act as a viral internet, constantly downloading and uploading genetic upgrades across the entire microbiome, bypassing the slow generational process of standard evolution.
This raises an incredibly profound question. If we synthesize all of this, if we combine the Thermocast9 epigenetic editors, the extracellular vesicle delivery networks, and the phage-mediated horizontal gene transfer, where does agricultural science go from here?
It goes somewhere completely unprecedented.
We aren't just breeding hardier corn plants anymore. The literature states we are shifting toward the design of holobionts.
A holobiont is a conceptual shift. It means we no longer view the plant as an individual organism. We view the plant and the billions of microbes living on its roots, in its tissues, and in the surrounding soil as a single, unified evolutionary unit.
We're engineering the whole package.
With the advent of AI-driven models capable of predicting complex protein interactions and the CRISPR tools to execute those designs, we can theoretically engineer the entire system simultaneously.
Imagine the possibilities based on everything we've discussed today. Imagine using Thermocast9 to program a specific bacillus strain in the soil so that the moment the IoT Laurelon sensors detect a drop in soil moisture, the bacteria automatically switches its epigenetic profile.
Oh, that would be amazing.
It begins mass-producing extracellular vesicles packed with highly engineered stress-tolerance enzymes and ships them directly into the roots of the corn plant to prevent wilting.
Or we return to the very beginning of our deep dive, the CN ratio. What if we engineer a strain of Clostridium that is completely immune to the rules of immobilization?
Wait, could we do that?
We could. We could program a genetic switch that bypasses the need for the 25-to-1 carbon fuel requirement. We could instruct the microbes to fix nitrogen from the atmosphere and pump it into the plant continuously, regardless of how much carbon is in the soil, effectively ending the microscopic bank run forever.
The potential is staggering. It is the absolute molecular-level optimization of the entire global nitrogen economy. We've traveled an immense distance today.
We really have.
We started at the macro level, looking at the seemingly mundane act of throwing a piece of cardboard and a banana peel into a plastic bucket. We drilled down into the strict physics of heat and the unforgiving 25-to-1 chemistry required to make that waste rot without volatilizing into ammonia gas.
Right. The fundamental physics.
Then we looked at how massive urban infrastructure is utilizing edge computing, 7-year LoRaWAN batteries, and thermal variance histograms to translate the invisible, smelly language of soil gases into real-time, actionable digital alerts.
We followed those gases to uncover the hidden nitrogen economy happening in the soil, exploring how the physical architecture of clay vaults dictates the battle between mineralization and immobilization.
And finally, we saw how the cutting edge of synthetic biology, from iron plaque electrical outlets to epigenetic CRISPR hacking, is giving us the tools to seize control of a 450-million-year-old biological engine.
We are rapidly transitioning from being passive observers of decay, constantly at the mercy of microbial bank tellers, to becoming active, intentional engineers of the global microbiome.
It fundamentally redefines our relationship with nature. There is no such thing as waste. There is only raw material waiting to be mineralized, immobilized, or entirely reprogrammed.
That's beautifully said.
Which leaves us with a final lingering thought for you to ponder long after this deep dive ends. If we are truly learning to manufacture and deploy extracellular vesicles as delivery trucks, if we are utilizing methylation-sensitive CRISPR to continuously rewrite the genetic code of soil microbes in real time, and if we are monitoring every microscopic pocket of heat, moisture, and methane with a globally deployed, edge-computed IoT network, will the farm of the future even resemble nature? Or are we slowly, steadily turning the entire agricultural surface of the Earth into a massive, perfectly tuned, globally networked cybernetic organism? The next time you walk out of the kitchen bin and toss a handful of vegetable scraps into the dark, just remember, you aren't just taking out the trash, you are feeding the machine.